Yeah. Está diciendo live.
Okay, so we can start ready, Fabien? Okay. Well, good afternoon, everyone. Welcome. It is today my pleasure to introduce Dr. Fabian Plison, who is giving the seminar today. His formal training, beginning as a bachelor's in life sciences, he got then a training at the graduate level in organic chemistry, chemical engineering, and biomolecular engineering. His work is, he has dedicated his research efforts to drug discovery, making an interesting combination between work in academic settings and interaction with industry in different universities, in University of Aberdeen, in collaboration with Aqua Farm Discovery in Scotland, CNRS in France, University of Queensland, Australia, and Spain as well, and also a collaboration with Pfizer. More specifically, his work is directed to the identification of bioactive natural products and drug design with a strong emphasis in peptides, using peptides scaffolds for drug design and screening. And he employs state-of-the-art analytical and computational tools to his efforts in drug design for diverse diseases. He has worked in efforts directed to looking for drugs for cancer, diabetes, neurodegeneration, etc. In 2017, he joined Langebio in Irapuato. He's almost our neighbor. And he started his own group, his own research group in the design of peptides for the therapeutical purposes. Welcome, Fabien, and thanks for being with us.
Thanks, Alfredo, for the kind introduction. So, hi, everyone. So today I will basically talk about the research I've been conducting with my lab over the last 3 years since I arrived in, started my labs in Irapuato, in Langebio. And so to start, basically what we basically interested when we build up protein design or anything like that, we are actually interested in the relationship that exists between a sequence, primary sequence, the structure that the protein can fold, and then the potential function associated with that structure. You've heard probably early this year, late last year, that there were tools applied in artificial intelligence, particularly to improve the understanding between the primary sequence and the relation to structure. So I'm talking mostly about AlphaFold from the DeepMind, the Google company. But today I will basically talk about the use of such tools to actually help to either understand or predict the function based on primary sequences, as well as to support the design of novel sequences that are not necessarily natural, and that we can use as a scaffold for potential drug applications. So my lab basically focused on 2 kinds of peptide sources. So on one side, you get the peptide known as antimicrobial peptides, the one that mostly interact with membranes, bacterial membranes. They also target intracellular proteins, but they're mostly known for these. And the other side, I'm also interested to derive and changes something that you may be more familiar with, toxins derived from venomous species that target ion channels and receptors, and that can be involved in a treatment like pain or epilepsy. Today, essentially, I will be talking about some of the research we've conducted around antimicrobial peptides. So as you may know, antimicrobial peptides are actually known as the innate line of defense against pathogens. They mostly associated with the interactions around bacterial membranes. They usually most likely the structure known for them are the random coil, and they fold into an alpha helix. They're also positively charged, which make them interacting electrostatic interaction with the negative charges of the phospholipids. And they can have a different mechanism of actions, mostly forming membrane disruptions and pore formation, responding to the lysis of the bacterial content. So these actual differences in mechanism of action led to consider that the antimicrobial peptide could be a new novel source of antibiotics. And so it's still considered today. Also, as you will see, they face a number of challenges to make them drugly available as a therapeutic treatment against infectious disease. Besides being interacting and disrupting the membranes, it is known more recently that a number of those peptides actually interact with the intracellular proteins. And here you get different targets: proteases, DNA, RNAs, the outer membranes, also the protein biosynthesis and metabolism. So those peptides tend to interact with this intracellular domain too, which means that there is not only one unique mechanism of action towards just membrane disruption. They could also interact internally with bacteria. Here's the type of interaction that is known to drive the antibacterial activity, mostly around the charges and the hydrophobicity characteristic of those peptides. Now, many of those peptides have been actually employed or are currently in clinical phases, mostly towards certain local and topical infections. And that's why most of them are actually applied as cream. One of the problems also that, and we'll see later, is that limits also the application is to do with that toxicity. So there's different kind of toxicity: cytotoxicity, hemotoxicity, and immunotoxicity. But we will try in this work to try to fix one of those problems, to resolve one of those problems by looking at their physical-chemical properties. And so, as you can see here, there is a number of applications. Another problem that has recently surged around antimicrobial peptides is also that bacteria start to develop as well phenomena of resistance against those peptides, which they haven't for a large number of years, but now it's mostly induced bacteria that have been associated with these kind of transformations. And but it still remain very little evidence around the bacterial resistance to those type of peptide. Another interesting information is that for their ability to bind to membranes, they're not necessarily selective towards those membranes. And what happened is that they could target as well the membranes of viruses. There is actually a number of peptides known to target SARS-CoV-1, not SARS-CoV-2, but SARS-CoV-1. They also able to target different type of cancers. And so they also been able to attract or use as a immunomodulator and call the innate and adaptive immune system for against potential infections. But beside all these fortunate properties, the antimicrobial peptide face a number of critical issues. One, as I mentioned, is it has to do with their high toxicity, which limit their application to skin and cream and topical application in general. The fact that they also not necessarily target specifically a number of those bacteria, but rather have a broad spectrum. Their functional promiscuity as well is kind of a challenge, because you don't want necessarily off-target issues associated with these peptides. And many of those peptides turn out to be alpha helices. Others could be rich in cysteines and present cyclic structure. The alpha helical and linear structure in general are usually unstable in change of thermal change of temperature and pH. They're also sensitive to proteolytic degradations, and they're likely not able to penetrate membranes and being orally bioavailable. Thanks. One of the way to resolve those problems in general in the labs to modify those peptides is to actually do protein design or protein engineering. And that's, as I've done synthesis myself, you end up choosing between different strategy, which is one could be the forming overlapping segment of the protein to further understand which residue are actually involved with the functions. You can do single mutation or double mutations around the sequence, like alanine scan, in order to understand which residue is part of the functions, truncation, and as well as positional scanning. And after all, when you do this kind of thing, you basically follow this kind of cycle. You start with a design, an actual data set of peptide or single peptides. Then you will actually develop a certain number of analogs when you will build structure-activity relationship. You test many of those peptides against one or several assays in order to associate a certain function with it. And then eventually you will decide based on a certain optimization which compound turns out to be your successful, your hit or lead structure. And I did just that when I was in Australia. I was working with David Craig and David Fairley, and this collaboration with Pfizer, where we're making analogs of GLP-1. So the protein that is associated with the GLP-1 receptor and participate to the secretion of insulin. I will not talk about that in that talk. Unfortunately, I will talk about other research I've done more recently. But so what I was doing there was mostly to constrain the structure with a number of bridges in order to capture the possible conformation that will be locked into the receptor itself. And then we were monitoring this type of information using the receptor agonistic, the peptide agonistic activity, the binding affinity, the capacity to secrete insulin or stimulate insulin secretions, as well as the capacity more physics to propense helical structure. And with this, we were able to build up certain relationship. And at the end, we had a winner, a double constrained peptide with a single mutation around what we call the kink, in order to get this sort of singular alpha helices, which showed some affinity and agonistic activity similar to the bioactive compounds in Byetta, so exenatide 4. But I mean, I was in Australia, there was a lot of resources, there was a lot of things, and we had the capacity to actually do this kind of experiment with no much worries around the time, the human resources, or the equipment. And so here I'm actually interested to reduce a bit the cost and try to work more strategically toward the insertions of automation when we can, and the use of machine learning algorithm and artificial intelligence to reduce some of those experiments and to learn further from what already has been done. And for that, that means aggregating information from the literature with a number of curation from this data, eventually put in place a number of models that are associated with either the prediction or generations of peptide sequences, and being able to automate the decisions associated with it. And so the cycle changes a little bit here. You start from either a single structure, a sequence or structure, or library of peptide. Then you will apply generative models such as de novo peptide design, which has been done for decades now. We can also implement deep mutational scanning, which has been mostly related to protein structure or binding affinity with proteins. We can also apply the more recent applications of neural networks, such as recurrent neural networks, GANs and VAE, to develop new novel sequence that look similar or that share similarity with the original sequences to actually build up and try novel sequences. Then you can monitor some of those new sequences by addressing like a list of fitness functions, so either one or several, as a proxy for the biological assay, and apply a number of filters in order to obtain your actual optimal sequence. So you could imagine here an IC50 of whatever biological activity. You get a single peptide originally that is red on the top in your fitness landscape. And what you're trying to do is to improve the biological activity as you make along different analogs. See, and then so the beauty about doing it computationally is that you are able to do more than one at a time, and you can monitor different biological activity in parallel, leading to the potential potent sequence that comply with most of those bioassays or in silico bioassay per se. But when you do machine learning, you not only need to just worry about the input data that you put in, but you also need to consider a number of parameters. For example, based on the size of the data set, you will either favor the use of algorithms such as in traditional machine learning, so that means super vector machine, random forest, linear regressions, if you talk about the regression models. Or eventually, if you are a large data set, you may eventually go towards deep learning approaches, such as the building of small or large neural networks. Sometimes, and that is actually true in a lot of biomedical data, we don't necessarily have a well-balanced data set and not complete data set. So you need to consider algorithms that take into consideration this imbalance. Also, experimentally, and then we saw that at least we heard about this kind of biases algorithmically with face recognition, but it also exists with the design of peptides. When basically you build up a data set that is biased towards faces, specific faces, or favor a certain subclass or variety of data, and then you end up having an actual bias toward a system of predictions. Finally, another challenge, and I will talk about that in halfway in the talk, the domain of applicability. So when the domain of applicability is basically telling you what are the limitations of your predictive models. Because many of those supervised algorithms, what they do is they're taking a labeled data set and then they build it from what we know from a given data set, a given database in time and a course of a certain time. So you will have information that is a state of knowledge based on what you collected. And that means that if you were to enter a new sequence, this sequence might be strikingly different to the others that you already had to predict your use. Even so, you may be able to predict a certain proxy and features for that. It is possible that your model is likely to extrapolate around this sequence and not give you a true and reliable predictions. So you need to take into consideration that here you can see this representation between the test and this overlap between a training set and a test set, so one in blue, one in red. So here they overlap perfectly, so there is no problems of domain applicability, but you can easily imagine that either one color or the other is outside that domain. So we'll first talk about the predictive modeling part. And the predictive modeling part, so in that case, we were interested to tackle and to resolve one of the problems associated with antimicrobial peptide. And I will talk essentially about the high toxicity. And when we talk about high toxicity, we refer to, in many cases, the activity associated with cationic antimicrobial peptides with erythrocytes. There is also cytotoxicity with other cell lines, but in that particular case, that is main emphasis around the toxicity of those peptides. And so what we did was basically to build up a model that could predict the actual hemolytic activity or non-hemolytic activity of a peptide based on these primary sequences. As well, and I would what you will see is the ability, because we build from possibly a given set of known sequence to be hemolytic or non-hemolytic, we also need to know the limitation and the diversity of those sequences. And so for that, it will eventually tell us the domain of applicability, and therefore whether our peptide is considered inside or outside that space of predictions. And then eventually we apply a certain filter. So here is basically what we started with. So in one, we basically build two sets of data sets. One will be used, the hemolytic data set that will be used to predict. They contain both hemolytic and non-hemolytic peptides. They also contained peptides that showed hemolytic activity at low and high concentrations. And so there's different type of data sets in that case. We also used testing sets. One was the 3,000 peptide from the Antimicrobial Peptide database. And now it's about 3,200, but at the moment, the moment of writing or doing these studies, it was around that figure. We also did an external validation using a known data set of antimicrobial peptide, hemolytic antimicrobial peptide. And finally, we generated de novo 5,000 random peptide sequences that share some the amino acids composition of antimicrobial peptides in order to understand further the relationship between sequence compositions and the predictions that you will see. So for each of those, because the sequences are quite different across the spectrum, you need to have vectors from the different lengths. And for that, you actually need to use physical-chemical descriptors as a proxy of those variety in sequences. Then after cleaning up the information, we first start by doing the actual classification models, which where the aim here is to actually evaluate whether our peptide will be hemolytic or non-hemolytic, or hemolytic at low or high concentrations. And so the results, we first start screening different binary classifiers. So that means it will tell you two responses, 0 or 1, and a class probability associated with each of those classes, which sum up to 1. And so here, what we evaluated different binary classifiers that in the first data set, what you see in bold is that 3 algorithms, gradient boosting, linear discriminant analysis, and XGBoost, were our best performers to separate the two classes. And in MOP2 and MOP3, also gradient boosting were also considered a good binary classifier. But the performance were lower because the separation was harder to make with those descriptors. Once we tune up each of the algorithm using their specific hyperparameters and using a number of reductions of the numbers of descriptors that are relevant or not for the classifications, we end up for the first models, which basically predict whether a peptide will be hemolytic or non-hemolytic, we were able to predict for any peptide sequences, primary sequences, between 95 and 97% accuracy. So that's relatively good. It turns out at the time of doing this information, there were already 2 papers that were published in 2016, 2017, and 3 that came out last year. And so we were actually interested to see what different can we bring, because our results were very similar to the one that those guys actually reported. So there was at least I was confident that the work we were doing was actually in the good directions. But what was actually interesting is that none of those papers actually identified the limitations of the predictive models. And for that, we decided to apply what we call outlayer detection methods. And for that, there is 2 kinds. You get the univariate approach when you actually reduce the data set to a single distance. And so that means you can actually draw an actual distributions, and will eventually establish that the data set of the dots at the extreme of the distribution will be your outliers. That the typical one used for reducing from multiple features to a single one is Malahon-Ibis distance. The second approach that we explored, and that was actually never reported in peptide design and rarely applied, is actually what we call multivariate outlayer detections. And these can actually be done based on either 2 dimensions, 2 features, or the 56 dimension we start with. So that actually helps us to reduce, to actually eventually identify the limitation and the boundaries of prediction of our peptide space, if you like. So that's actually features like that. You get different algorithm. And so you could see here that, for example, the majority of our peptide will be these white dots, and the black dot around it will be the outlier, the very special cases, and there is not enough distribution to consider them in layers per se or part of the main distributions. And depending on the algorithm you use, you either count them in as in layers or you count them out. And so what you see here is that the shade of blue basically reflects the whiter it is and it's basically more proven to be reliable. And the further away you are from this orange zone, if you like, the further away you basically, your prediction will be less reliable, and your outlier will be actually considered as a true outlier and couldn't be used for prediction approach. And so for what we did, so that you see on the right part of this exercise, these slides, we actually compared the different models and benchmark them in order to see which method will be the most appropriate to identify the limitation of our first models, which is MOP1, so based 95-97% accuracy. And for that, what we were trying to do was consider that both the models dataset that I used to train the model and as well our external datasets were true non-hemolytic or hemolytic peptides. So they basically represent the maximum space that we can actually determine. So what we were interested to do here was to identify this sort of red dashed boundary, the learned decisions that can encompass the majority of those peptides. And for that, so that's that will be the famous white dot. And so here we tried different ones, and what you see in a dark color, so either red or green, will be the actual percentage of outlier or novelty associated with it. So we try to minimize those values. We by looking at these plots, basically what we see is that average KNN, which is one of those methods, will be the best way to detect those boundaries and encompass the majority of the known peptides. And so that's basically, it turns out to be the same algorithm to work for all the other two type, MOP2 and MOP3 datasets, different values. So. And so what you see here is that's the system works. Average KNN will actually identify certain islands with low probability to be an outlier or high probability to be an outlier. What you see in the middle of the slides is basically that you get the training set here, MOP1, that is displayed from 56 dimension to 2. And I use t-SNE as one method to reduce the dimension and to be able to visualize the two classes. In purple will be the classes that present the peptide that present high hemolytic toxicity, and in orange brown will be the one that shows very low hemotoxicity. When we apply that model to our training, our testing set, the IPD, you can see also here different occupation of the peptide space, but you can also see that there will be a number of those peptides predicted to be purple, so that means hemolytic, and a number of them will be orange or brown, being non-hemolytic. When we apply this average KNN to both of those data sets, what you see below in a shade of red is basically all the one light red will be considered in layer dataset, and the one in dark red will be the outlier, the one outside those famous black dots in that picture. So that means here we try to minimize the peptide, and then most of those black dots, the red dots, are actually found in the extremity of the space. So they usually, as you can see here, there's very little of them. When we apply that same algorithm to our test set, you see that a number of dark blue dots represent the outlier that will be associated with the testing set. And so I would say, so here now we were interested to actually apply that to our predictive model, both the detection outlier and the prediction per se, to our testing set. I show you before an example with the APD, but so you can see another projection here. When you basically see that on the y-axis, you get the probability of each of those peptides to be considered hemolytic, one being the most hemolytic. And on the x-axis is the peptide that are likely to be outside, beyond one, is the one being outside the actual space defined by the training set. And so what you see here is that 70% of the antimicrobial peptides were predicted to be hemolytic, which kind of goes along with what it's usually say in the literature. Also, there was no number identified, no quantification per se, or in the literature until now. And then there is also this 273 sequences That are actually those dark blue sequences on the right side of the plot, which are basically hard to predict properly, and basically the prediction are not to be trusted. Only for those, not the one inside. And so we also apply that to a different type of families, including peptides that are actually reported for hemolytic activity. And so here you get different families reported with those values. And basically we're able to predict 300 out of 317 hemolytic peptides to be correctly assigned as hemolytic or predicted to be hemolytic. So 95% correct. Now, where is it? Up. Now, if you're not familiar or you're not interested to apply directly machine learning to your own data, or you don't have the expertise, well, we are currently developing this actual platform called AppMode for hemolytic activity prediction with multivariate outlier detection. If you have any more trendy name, I'm open to listen. I didn't have an idea so far, but basically what we're able to do is you will be able to submit your FASTA file in a normal manner, and then the website will basically return you the complete predictions for each of the single model. Actually, in the original paper, there is 9 models, because it's often considered that rather than selecting one single model, you take maybe a couple or 3 of them, and then you use the consensus approach to not favor one approach over the others. And that's particularly true in our method because they were very close to one another. If you get one particular algorithm, maybe you don't need to do this. But basically, you got this as well as the outlier prediction associated with each of the peptides. So here in blue at the bottom, basically is a result from the training set. And in the red dot correspond to your FASTA file, the 5 peptides that we predicted to be either hemolytic or not. And so this first phase of the work, basically around predictive modeling, show you that we were able to predict the hemolytic nature of any peptide sequences. We're able as well to validate them externally, that many our models were able to predict externally without a set of non-hemolytic peptide. We also applied the multivariate outlier detection for the first time to the design or the predictions of peptide activity. And I'll show you the example of these applications in the entire antimicrobial peptide database. Now, the question is, can we use these models as part of a design approach, not just the discovery? Until now, we just discovered the antimicrobial peptide that could be the 30% antimicrobial peptide that could actually be potentially non-hemolytic. And so here to actually talk about design, we need to consider the second approach in machine learning, which is generative modeling. And generative modeling is the idea that we can actually start from a certain type of a data set, and we learn from the data set a number of information, which is represented here as a noise. And from that, we can reconstitute a number of faces, in that case, faces or peptide sequences from that noise that share the same properties and the underlying distributions from the sequence that we used to generate those sequences. And so in general, that can be displayed this way. So you get a true data set in blue that shares image space. In that case, that could be peptide sequences. And then you actually apply a certain type of generative algorithm. There's different type in order to reproduce and to be able to generate a distribution that will be similar to the one that you used initially. And with this, you can actually do fun application. You can make yourself all look older with ASAP. You can actually generate the faces of people that do not exist. Or you can actually add yourself glasses that were not there initially. You can also create text that has no human input, that is as the structure of an actual sentence that will be in any kind of book and will actually make sense and is readable. You can actually see the original article in the Guardians if you like to read it. I didn't put the link here. But you can also apply that more closely to protein and peptides using, in that example, they use GANs, but in 2018, there was already papers publishing around using LSTM-RNN, different type of machine learning and neural network. And so if we go back to our cycle of design, so we start from a single or different peptides known to be hemolytic or non-hemolytic peptide, for example. Then we apply a generative model, as I said, to be able to generate from the noise a new sequences. And this noise can actually be the amino acid composition per se. You then apply your predictive model to see whether whatever you use in the first place can actually be used to filter out the sequences that do not fall into what you like, meaning non-hemolytic sequences, for example. And so one application that we actually incorporated into the AppMod API is actually the basically copying around the alanine scanning library. So that actually called deep unilational scanning. Deep unilational scanning is the idea that you generate single or multiple mutant from a single peptide sequence, a protein sequence, and then you see how those mutations will be affecting the fitness function of interest. So it has been published since 2014. One example is the binding affinity of a ligand to a protein, the stability of a protein, and more recently, the lab of Deborah Marks applied double mutants to predict, to use double mutant to predict 3D protein structure. So the idea here is basically you submit a single sequence at a time, and you came up with this kind of plots. The heat map basically will tell you on the top here, the columns represent the sequence of a peptide known as brevenin 2 related peptide, the peptide known to be hemolytic as well as anti-cancer. And then we, each row represents a single mutation applied to each of the positions. And what you see in the color basically are the probability of being hemolytic to be monitored. And for example, what you see here is that by inserting either an aspartic acid or glutamic acid at certain position, we pass from an orange value, which is the original value of these peptides, to a dark blue or blue value, meaning a very low probability to be hemolytic. You can also see that the insertion of lysine will be the opposite thing and will actually support and increase the hemolytic capacity or at least predictions of this peptide. You can monitor as well, because we were talking about domain applicability. You generate novel sequences, whether those sequences will actually be outside the scope of prediction as well. So you can actually apply that same thing, but different functions here, the probability to be hemolytic to that same data sets. And here on the other side is the same as the same heat map, but I just measure the differences between the peptide of references and the resulting peptide, the query peptide. So that it's clearer that the effect that, for example, the aspartic or glutamic acid have over the probabilities, you reduce by 0.6%. Now, another application, didn't change that value, was to actually generate de novo 5,000 random peptide sequences. And the idea here was to actually trying to understand what the amino composition was of each of those thousands peptides, and how does that affect the actual composition in both positively charged and negatively charged residue, as well as, so what you see on the left side is basically the probabilities of each of those 5,000 peptides, but either charged with a high frequency in glutamic acid or aspartic acid on one side, so they will be orange to brown, and they tend to have probability to be non-hemolytic because they below below that value, and then peptides are usually loaded with positively charged residues will be higher up in the probability predictions. On the other side, if you actually look at the amino acid content, but this time aliphatic and aromatic residue versus a small residue such as glycine, cysteine, alanine, proline, and serine, then you actually see that those small residue tend to push the presence of a small residue tend to push the prediction towards a low outlayer outside the boundaries of prediction. So that basically tells us that amino acid with small amino acids are unlikely to be predicted correctly. A final application of that predictive model is when we actually try to design anti-cancer peptides. We identify a number of peptides, and we identify that a number of them could be actually selective between different cell lines. And so what we were interested to do is to eventually design sequences that could be highly parented to one another, and one could be actually targeting selectively cancer cell line, and the other one will not, will be more broad anti-cancer peptide. And so what my students, Christiana, did was to actually start from 140,000 peptide between 15 and 25 residues. Then we applied different filters first to detect if each of the sequences will be considered an anti-cancer peptide or not, whether we can actually see whether the peptide will be a non-hemolytic. We are actually trying to do non-hemolytic peptides, so we pursue these directions. And so what we were trying to do here, she designed a binary model that could actually predict between a selectivity between an anti-cancer peptide between one or different cancer cell line. And so she developed these models. She applied it to her what's left from 1,400 peptides, and ended up with 6 peptides that are highly similar between one another, at least in pairs. And those are peptides here. And so right now we're actually collaborating with my ex-colleagues in Australia to synthesize those peptides and test them in different cancer cell lines. If you ask me about the different cancer cell lines, it turns out that the model we used to predict the anti-cancer activity are highly biased towards hepatocytes and other type of cancer cell lines, and we expect that the response will likely be toward those cell lines. And so I hope here I was able to show you that with generative model, we are able to not only amplify the actual characteristic that make a non-hemolytic peptide. You saw that that, for example, with hydrophobic residue and the combination of hydrophobic and negatively charged residue, we're likely to get or predicted non-hemolytic peptides. We also applied this model to filter out the design of non-hemolytic selective anti-cancer peptides, and also show you how we can apply that to, for example, peptides that have very low homologies, and you need to do single mutants or double mutants or more against a single sequence to measure and potentiate the actual mutations. And without further ado, so that's the actual my lab right now. So 2 of them are no longer there, but that's my lab IDs. We still continue to work on Zoom. Some of them are back in the laboratory. We still work at 20–30% as well. And that's it for me.
Okay, thank you, Fabien, for a very interesting presentation. We have some questions. If you have questions, please type them in the Q&A section, and I'll read them for you, or you can ask them directly. We also have 2 questions. One comment from Juan Pablo Robles, very interesting hypothesis and results. The computational peptide design is mostly focused on the structure parameters. And the question is there some algorithm that could predict toxicity, half-life, potency, or other functional parameters for those peptides? How difficult is that, and how difficult is that?
So right now, most of the predictive algorithms that I show you are not actually focused on structure, because for most of those peptides, the structure is actually not known. So we can predict the secondary structure. We can eventually I'm likely to say that many of those peptides are alpha helical, but we don't know the actual structure of those peptides. Most of the predictions are done from primary sequences. Now, toxicity, well, that's one example to measure toxicity. There is also predictive models that are associated with cytotoxicity and immunotoxicity. Half-life, well, that's a bit more complicated because you will have to actually have a lab with a lot of data and a set type of model, whether it's a rat or mouse, or whatever you want to use to measure this half-life. So it will be very noisy eventually. So on top of my head, I actually don't know any models towards that. But yeah, maybe there will be an interest if you get a solid number of data set around that with the same type of animals you can use. Happy to develop an actual model for it. And the last one was another.
Is how difficult is that? How difficult is this to? Well, is you already answered related to. But there's another comment from Juan Pablo as well. He says that many therapeutic peptides use specific modifications such as modified amino acids. Do you consider relevant to include these possible variations on the predictions? The use of
the one I actually
modified
use,
amino acids.
Yes, actually, in one of the slides I showed, there was one of the groups that developed predictive modeling where they used chemically modified peptide. So that means they didn't have not only the non-proteogenic amino acid, but they also had all. Yes, it's possible as long as you have sufficient variants, so sufficient analogs to consider each of those mutation relevant. I mean, if you only have one mutation on one single peptide out of 2,000, it will do nothing. So it basically will be eliminated because it doesn't have much weight in the predictions. In that case, we only use the 20 amino acid predictions. But yes, I mean, as long as we can codify each of the different mutations, so a non-proteogenic amino acid into a code, whether it's 1 to 20 or 1 to 100, well, you can actually predict, do prediction this way.
Okay, thanks. And another general question is why most peptides that are in clinical use are applied topically? Why is that?
So one of the explanation I had and I explained was that many of them are actually hemolytic. So unless you want to actually have an hemorrhages, you likely not to apply that other than the thick skin will avoid to the set of side effects. That's one reason why they actually limited to topical applications on skin at the moment.
Okay, and a question in a way related to what Juan Pablo was asking before. How do you go about looking for new peptides related to a specific new effect? For example, you have the effect, the biological effect, and you have the protein that does that exerts that effect. How do you go about on this generative modeling to identify new variants, or peptides, better peptides that would reproduce that effect? What information would you need to do that modeling?
Well, so you can actually do generative modeling. You can do that either by having an heterogeneous data sets of peptide like antimicrobial peptide, or you can have highly relevant or highly structurally related peptide like the venom toxins. And so what we're interested to do is to use a certain type of algorithm, LSTM-RNN, variational autoencoder, etc., to generate novel sequences, even consider having to merge two protein families that actually targeting the same ion channel, for example. And you can actually element that to feed up and to generate novel protein and scaffold possibly.
But if I have the function, let's say the effect of a protein is a completely different to anything that has been modeled before. It's a new thing. All the information that I have is that that peptide exerts that biological effect. Would I need additional information that is and other proteins that do the same thing, or maybe try variants in the laboratory before the modeling? Or
yes, I think in that case, likely to do what's called the. So if you are able to predict that biology. If we're talking about, you can either do mutation in a lab. I mean, you can do this. If you don't have much information, if you are able to do a predictive modeling on the other side based on a diversity of sequences, not only the sequence that you have, then you can do what I've done with Breblin type 2. You can actually have your sequence and then do single mutation, predict the actual mutation, and see the effect that it could have every single one of them. That could be protein stability, that could be ligand affinity, etc., etc. So you can actually either do. There's two areas that we're currently developing. One is the forward approach, that is mutational this way, and the other one is inspired from Frances Arnold's work around machine learning directed evolutions, when we are basically trying to use large families of peptides, and from that we are able to identify the key residue associated with a single function and the weight that each residue have with that functions. So there is different ways to actually explore this approach.
And as an extension of that, is your group in a possibility of developing collaborations with people working in the neurosciences, for example?
Yes, I mean, I'm currently doing, we're exploring, some of my students doing work with scorpion toxins, ant toxins, and conotoxins, which we know that targets ion channels. So I don't have any ion channel. We use sodium ion channel at the moment for one of the target, and acetylcholinesterase as another target. But yes, I mean, we definitely can actually focus on a specific peptide ligands and look at how we can apply some of the machine learning approach to either understand the residue associated with the interactions, or to develop new analogs completely different to the ligands. If that's what you're trying to do, is, I don't know, intellectual properties, to develop new the same ligand with that conserve the structure but without having the same sequence. Anything like that can actually be can be done. Yes, I mean, right now we're just starting on ion channels, ion channel and conotoxins and all that. So but yeah, definitely neurodegenerative disorder. Actually, I did my PhD with Alzheimer's disease and my postdoc with type 2 diabetes. So somehow I was a bit in the area.
Okay, because I see several possibilities of interaction with people in our institute. Well, if there are no more questions, there is a question by Manuel. Manuel raised his hand. Go ahead, Manuel.
Okay, thank you. Hemolytic peptides can be identified by assays based, for example, on sheep erythrocytes, or even human erythrocytes, or other species, erythrocytes from other species. Do you take into account this bioassay to identify hemolytic peptides for some step of your classification or modeling work?
Right. Thanks, Manuel, for that action. In that particular case, the curated database we use for training set was actually built and curated from human erythrocyte mostly. I know there is actually information about the hemolytic activity associated with the different species, as well as the different rhesus associated blood type will actually could influence the biological activity or the hemotoxicity of a single peptide. So in that case, I don't think there's enough information about the blood type, but there's definitely information about the origins of the erythrocytes.
Oh, thank you. And another question is, what can be expected about the immunogenic properties of these design peptides if applied orally, for example?
Actually, I don't know. I don't know in that area. I haven't explored too much on this immunogenicity at the moment.
Okay. Thank you very much. Congratulations.
Thanks a lot.
Okay, well, I don't see other questions. I think I don't know if I see all of them, but if there are no more questions, thank Fabien for this interesting presentation, and maybe some interactions could develop with the Institute.
Thanks a lot. Thanks a lot for the invitation again.
Thank you for being with us.
Hopefully soon we will be able to see each other physically.
Okay.
Querétaro is just not far, not really far.
That happens soon. And thanks everyone for tuning in, and we'll see you next week, or I don't know, in the next seminar. See you, Fabien. Thank you everyone.